Thank you for coming to this session, which is, the last talk in the summary session. I'm going to give a summary of the cost session from yesterday, where we had several different speakers. I don't believe any of them are here right now. Yes. First of all, Juliana gave an introduction about our cost action, which is the European network, where we have over 30 countries now that are collaborating, communicating to advance the science and medical care for aniridia, and also we have a number of activities which are designed to train the next generation of researchers and clinicians to understand more about the very particular circumstances of aniridia. So we have many different events being organized, like scientific sessions and talks in international conferences and training schools and scientific exchanges between countries. So then, after my summary, Professor Dominique Brémond-Gignac from Paris, from Necker Hospital in Paris, gave a talk, and she's a pediatric ophthalmologist there, and also part of the ERNI network, and also part of our cost network. And her talk was about foveal hypoplasia. Of course, we know that this is important, foveal hypoplasia, because it's affecting well over 90, probably 95% of people with aniridia have some degree of foveal hypoplasia. Often it's difficult to assess because of the nystagmus and partial opacity in the cornea or the lens makes it difficult to see to the back of the eye to perform a proper OCT examination. However, despite this, Dominique's team was able to examine 111 eyes of people with aniridia. 111 people and 205 eyes with OCT. So this is a very large cohort, a very large group. From which we can draw important conclusions. First of all, it's interesting and important to look at the foveal hypoplasia because it's very strongly correlated to visual acuity. However, we should also keep in mind that an absence of a fovea does not necessarily have a major impact on visual function. It can, but it doesn't need to have a major. So that's important, but it is strongly correlated to the visual acuity. There's a grading scale of foveal hypoplasia, where 0 is a normal fovea. So you have a normal foveal dip, where you have, for sharp vision, where you have the greatest concentration of the photoreceptors in the retina. But then we have other grades where this foveal pit, it's called, is less and less visible. So the grades go from 1 to 4, where 4 is just basically a flat foveal region. This is also often combined with vessels, vascular invasion of the fovea, which normally should not have any vessels. And also, this is pigmented region of the retina, but often when we have these higher grades of foveal hypoplasia, that there is low pigmentation in that region as well. So from their cohort, they could say that 73% of those 205 eyes had the most severe grade of foveal hypoplasia, which is grade 4, while the other grades, the more milder grades, were about 5 to 8% of the subjects, of the eyes, having those milder grades. So really, overwhelmingly, it's a grade 4 foveal hypoplasia. However, it doesn't necessarily need to be the case. There was about 6.5% had a grade 0 foveal hypoplasia. So this is like a normal morphology of the fovea. And also, it's important to keep in mind that it doesn't have to be the same in the left and the right eye. The grading can be different depending on the eye, so there is some asymmetry. And also important to note is that although overwhelmingly it's a grade 4, 73% had a grade 4, this is very much dependent on the specific genetic mutation in PAX6. So it turns out that if it's a coding mutation that affects PAX6, so coding for the protein, then in 93% of the cases, it is a severe grade 3 to 4 hypoplasia. However, if the mutation is in a non-coding region, it tends to be a 0 to 2 grade of hypoplasia. So really, this is strong evidence this is really genetically determined. I think also what's interesting is we see the same pattern in the keratopathy as well, that when we have the mutations that are not affecting the coding for the PAX6 protein, then we have a mild keratopathy as well. And it's the same pattern. And also when we have the coding mutations, they tend to be more progressive or more severe. So this is the same pattern that we see with the foveal hypoplasia and the genetics as well. Dominique's group also looked at the iris hypoplasia because, of course, this is a very visible feature. And they looked at partial iris hypoplasia versus total. So total meaning iris completely missing. And there they found that about, if there was an anomaly in the iris, so partially missing or up to totally missing, then 50% of those cases had the grade 4, the most severe foveal hypoplasia, whereas if it was in the other half of the cases, it was less. So I think the message there is that the iris hypoplasia tells us less about what's happening in the fovea than the genetics. The genetics is really more important there. So after that talk, we had Professor Daniel Aberdam from INSERM in Paris, where he has a research lab there. He told us about limbal stem cells and also the role of PAX6, not only in limbal stem cells, but in the cornea, and also talked about drug repurposing for the keratopathy and aniridia. So this is important because, of course, the keratopathy and aniridia has very few successful treatment options. So there's the surgical options, but as we heard this morning, many of those are eventually not successful. So the goal from the research standpoint is to not get to that point, but to have some kind of treatment, develop some kind of treatment where that keratopathy can be delayed or prevented from happening. And so what's interesting is that Daniel Aberdam's group has developed a model for, and we know that the limbal stem cells are very important and their status is very important for keeping the cornea clear. So what Daniel's group did was they took limbal stem cells and they modified with this CRISPR technique, they modified one of the PAX6 alleles to make an aniridia type of stem cell. So this is important because we can't really take limbal stem cells from people with aniridia because you would have to take it at a young age and so forth, and of course that's invasive. And it would trigger the keratopathy. So it's important to do this in vitro in a cell model. So they were able to do this. So we have cells that look like the limbal stem cells from aniridia. And what he used there was he used those cells to screen a whole library of over 1,000 drugs that are already approved drugs. And so by doing that, they could figure out which of these drugs could actually increase the production of PAX6 protein in these limbal cells that had the aniridia mutation. So what was interesting there is that a few drugs came out as positive from this screen, and these are the two drugs, mainly the two drugs that he mentioned were called duloxetine and ritanserin, and these are both antidepressive drugs. So they are increasing the amount of norepinephrine and serotonin that is present, and this has an effect that it actually activates PAX6 to the gene to increase the production of PAX6 protein. So it's also interesting that not only Daniel's group has shown this now that this works in cells, but a group of Ali Jalilian in Chicago has shown in a mouse model of aniridia that the same pathway that these drugs affect can be modulated by another type of drug, which is called a MEK inhibitor, and that could also increase the amount of PAX6 in the cornea, and in the mice give a better, more transparent cornea, so delay the keratopathy. So that's very interesting. However, the work in the United States is using a MEK inhibitor, which is not an approved drug. It's in trials for cancer. Whereas Daniel's, the drugs that, the duloxetine in specifically is an approved drug, and it's quite a cheap drug. Also, Daniel talked about inflammation, and this morning we also heard about inflammation and how important inflammation is to consider in the cornea, in the keratopathy. In particular, we heard that the inflammation occurs probably before, or most likely before, the limbal stem cells become insufficient or deficient. Let me see. Daniel also posed the question whether the immune cells, so the inflammation we see, those immune cells, could they be affected by the PAX6 mutation? And he talked specifically about one type of cell, which is called a microglial cell. It's a type of nerve cell. And this strongly expresses PAX6, but only shortly after birth. And then the expression decreases. And these microglia are present in the retina, and these type of cells are also present in the cornea. And depending on their environment, they can either result in protecting the nerves in the cornea or degenerating those nerves. So if it's an inflammatory environment, then these microglia will release factors and the nerves can degenerate, which is of course bad because it's harder than for the limbal stem cells to be maintained. If the environment, however, is non-inflammatory, so when these microglia are producing PAX6, then it stops the inflammation and protects the nerves. So it turns out that duloxetine can actually increase the level of PAX6 produced by these microglia, thereby promoting a anti-inflammatory environment and hopefully protecting the nerves. So this is the hypothesis. And Daniel just raised the question whether inflammation is a primary cause of aniridia-associated keratopathy, whether it's rather inflammation and not the limbal stem cell deficiency that's causing this. And we have several sort of observations and lines of evidence that seem to support that hypothesis. And then the question becomes, what is really the function of PAX6 in inflammation? We know that in the keratopathy, the cornea is in like a chronic wounded state where PAX6 is deficient. So there seems to be some kind of a direct connection there. However, the research is ongoing. Then we had a talk by Heli Scottman, Professor Heli Scottman from Tampere University in Finland. And She is an expert on stem cells, stem cell culture, and in particular she talked about iPSCs, or induced pluripotent stem cells. So now this is a very interesting technology that has arisen within the last decade. It really started from Yamanaka, who was a Japanese researcher who won the Nobel Prize for his discovery that we could take adult cells and turn back the clock on these cells and make them like a stem cell early in development. And why is that important? Well, that's important because if you have this stem cell early in development, then that cell can be made to create the cells of various different tissues. So it's pluripotent. It can create different stem cells, including the limbal stem cells. So this is what Hallie was focusing on, using patient-derived cells and then you modify 4 genes, and then it basically transforms the cell back to this pluripotent stem-like state. And then what they do is they add certain factors in vitro, in culture, and they differentiate those to the limbal, just to the specific limbal stem cells. The good thing about this is that those would be autologous stem cells. They're coming from the patient themselves, because you can't take it from the other eye, because both eyes are affected. So this would be, in the future, this could be very interesting type of therapy, because it wouldn't require the immunosuppression that you would need if you had donor cells. However, more in the short term, I think it's important to use this technology to learn more about how limbal stem cells derived from people with aniridia behave, and what can go wrong in their function over time. And by understanding that better, perhaps we can even in the future develop some therapies to maintain that stem cell function for as long as possible. So Hallie showed us that when inducing this differentiation to limbal stem cells, that different subpopulations of cells come at different times on the way to limbal stem cells, and they're trying to understand this whole process of how these limbal stem cells are created, doing sequencing studies. Also, these limbal stem cells that were derived from the patient cells, they seem to look like normal, healthy limbal stem cells, even though they are producing less of the PAX6 protein. Their other features seem to be normal, which could actually support the hypothesis that we have that actually the limbal stem cells in aniridia are normal at a young age. And which also, which we can see when we look at the cornea, that it is transparent for a number of years before the keratopathy starts. So this is very interesting. Even the wound healing that can be driven by these cells can be normal. So if these patient-derived cells made into limbal stem cells seem to be normal, then what does that mean? Why does the keratopathy develop? Why does the limbal stem cell deficiency develop? Well, we talked a little bit about potentially it's maybe not the cells themselves, but it's in the environment that the cells find themselves in. And so this needs to be looked at in more detail, and inflammation, of course, is one of those factors that can radically alter the environment of these stem cells. Finally, I think it's a very interesting new area, which is bioprinting that Hallie talked about, that they are starting to now develop small models of the whole cornea by bioprinting the tissue, including cells and nerves into that tissue. And this is a really great type of model that we can use in the lab to better understand how the different cell types interact it's better than just culturing one type of cell in vitro. So that is something for the future, using these organ-like models. And then finally, we had Dulce Limakuna, who is from Zhou Zhao's group in the Netherlands at Radboud University. And she talked about also about repurposing read-through, what are called read-through drugs. So the drugs that Daniel talked about, which is duloxetine, that is a drug that could be used on all in theory, on all mutations of PAX6. So it can be used on the premature stop codon mutations, but it can be used on missense mutations and other mutations as well. So, and that's because that drug works on the healthy copy of the PAX6. So we have the two alleles, right? We have the two copies of the PAX6 gene, and it works on the healthy copy of the gene, so that overproduces PAX6 to compensate for the other copy that's not producing PAX6 protein. By contrast, these read-through drugs like adalurin, you know about adalurin, the read-through drugs, they work on the damaged or the mutated copy of the gene and trying to fix that premature stop codon or ignore it to then produce the full-length protein. Because of that, those drugs, the read-through drugs, will only work on the premature stop codon mutations. However, that's about 70% of all people with aniridia have that type of mutation. But it's important to realize that it's not all the mutations. But nevertheless, she talked about these are nonsense mutations, and the drugs like adalurin are suppressing this nonsense mutation. But it's important because to realize that the drug, the adalurin, it's been around for a while, but there are also a newer class of read-through drugs that are potentially more potent and could be better than adalurin. And she has tested some of these drugs, and in particular, one drug called Amleximox. Do I pronounce it right? Amlexinox. She has tested on two models, one where these pluripotent stem cells derived from someone with aniridia have been, again, like Helly did, differentiate them to limbal stem cells, and then she tested this drug, Amlexinox, and it seemed to rescue the PAX6 production. So it did indeed compensate for this stop codon and cause the mutated gene to produce more PAX6. So that's good. They also showed in a organoid model where they used those stem cells to create like a mini corn or a mini eye. It was an optic cup. They also could induce that optic cup to produce more PAX6 by giving this drug. So this could be a promising candidate for the limbal stem cell deficiency in aniridia. Also because Amlexinox is, like duloxetine, an already approved drug. And perhaps, I was talking with Dulce afterwards, perhaps this has also already been used in the eye even, and potentially even as an eye drop formulation, which would make it even easier or quicker to approve. However, these are just in vitro studies, and it needs to be shown that this has an effect in animal models, small animal models, like a mouse model first. I think the discussion with Elizabeth Simpson was very relevant with their models, that they have very good models that this could be tested on, and I hope that could be a good collaboration to bring this forward into animal models. If it would show a good effect in animal models, then one could think about applying for some first human trials, like they did with adalurin. It's also important to state that even with the human trials for these kind of drugs, it's really important to have a proper design of the trial so that the trial is actually including people who could benefit from this, meaning that maybe the keratopathy should not have gone so far, so they should likely be young people, and a proper dosing also has to be considered. How often should it be given? And the form, topical, is probably the best for the eye. The form it should be given, and for how long it should be given, and also what are the outcome measures that you are going to measure to see if the drug actually worked. These are really important parameters to consider because, for example, ataluren could be actually an effective drug, but the way that it was evaluated could unfortunately lead to the wrong conclusion that it may not be an effective drug. So this is very important to consider as well. Yes, so that was basically the session from yesterday, and we'll take any questions if you have any. Anyone?
You have such a great talent to explain difficult things by a very simple way, and then all questions. For example, I got yesterday. You explained so clearly today. Yes, I just want to thank you.
Thank you.
For what you did now.
I hope I understood the speakers and did them justice.
I support that.
Okay.
So wonderful talent.
Thank you.
I'd echo what Galina just said. Thank you for a very clear explanation of all that. Just a clarification, you drew a distinction between visual acuity and visual function, and I'd like to understand what the difference is, and particularly what you meant by visual function.
Yes, so visual function meaning that you can perform real tasks or necessary tasks. So you may not have a high visual acuity, meaning that you see everything as sharp as possible, but you have enough vision to be able to navigate, for example, or to recognize a face, for example. So there is a distinction between the functional vision and the vision that we measure on a chart, for example. Yes. And I think that's also a very good point, because a lot of studies that we do, scientific studies that we do, there's an overwhelming focus on the visual acuity, so the numeric vision. But I think we need to be better actually at looking at the functional vision and reporting outcomes of functional vision. Because at the end of the day, that's what matters to you, not what number you have, but how you can function.
Just to add to this last point on visual acuity versus visual function, I think it is indeed important, because I've heard a couple of times that the eye has been given up because there's a known rather small visual acuity, but this is at least my perception that anything you have left, you should really value it. Then a question, you were mentioning CRISPR. I'm not sure if I missed that in previous conferences, but at least it's now popping up here. So my question is, one, how much hope should we put on the CRISPR technology for aniridia? And two, as I'm coming from a computer science background, and AI is now, let's say, kind of a hype. How much is CRISPR in biotech a hype?
Okay. Thank you for the question. I'm not the expert on this. Perhaps others can jump in, but I should say that CRISPR is a technique that works. It may not be the most efficient technique. There are newer techniques even than CRISPR that have been developed, like the base editing we heard about, for example. But CRISPR is a technique that works, and it's very valuable definitely in the laboratory, and when we look at in vitro studies and even small animal studies to understand what is happening at the biochemical level, and whether we can influence that by making changes in the genes. So for our knowledge and for our understanding, it's very important, and it's going to be very influential in the coming years. When we talk about therapy, however, there's a big gap from using this in a lab to actually having an approved therapy with a CRISPR-type technology. So there's a large hurdle there, and I don't think that's, in my opinion, it's not going to be solved in any time soon. It's going to take years and years. That has also been set back by there was obviously ethical issues as well, and that's been set back by a well-known example of a researcher who actually applied CRISPR in germline cells to actually that went to term to pregnancy to actually create. There are individuals born that were CRISPR edited, that has really raised the ethical debate and also probably raised the bar for what's needed to actually try to approve this kind of technology. Elizabeth?
Hi, it's a great question, and I agree with everything Neil said, but I would take a little bit more positive spin. I mean, the FDA has 33 ongoing CRISPR clinical trials right now. 7 of them are in vivo, and 1 of them is for Leber's congenital amaurosis, number 10. So I think things are a little bit more hopeful. I still think years is appropriate, but for some of this ones that are in clinical trial now, they've reported on the first 2 patients for the Leber's, and they so far are doing well, early days. But so I think it is coming, but still years for aniridia, I would say. Yeah.
Great, thank you. I've learned something. That's great.
I was, if I understood, there are some patients that do code a protein, but it's modified, and that's a small amount of patients. How does that, is it different in terms of how it affects the possible keratopathy, etc., and is there any research being done on what these modified different proteins do? Because I understand that if they have a different protein, maybe, but they still have a lack of the one that they're supposed to have.
Well, so if we're talking about the premature stop codon, they are producing a truncated protein which is believed to be non-functional and also subject to decay mechanisms, so that it wouldn't really last very long in the eye for those proteins. But of course, I'm not the geneticist. I think the genetic people with genetics background could answer that better. However, I do know that there's not much being done in terms of relating residual modified protein function to phenotype. So I don't know if anyone else knows about that, but one could hypothesize that if there's a partially functional protein, that it could correlate with, there could be like a scale where if you have a partially functional protein, then it could could lead to perhaps a milder phenotype, even though it's not completely functional. But again, I don't know if any studies are really looking at that. If anyone knows.
Thank you.
Yeah.
Good morning. I'm a geneticist, so maybe I can quite respond to your question. And as Professor said, when we are in front of a mutation that is a nonsense and frame shift, the protein is not completely in its entireness. But we also saw that there are missense mutation when the protein is complete itself, but the function is completely different, and you can have the pathology for sure. So what we are trying to do is to study, first of all, in silico models, and maybe I can answer to the question of the person before. We are starting to predict the function, and then with in vitro models, we are trying to simulate what is the deleterious effect when the protein doesn't work well, because the protein is complete, but it doesn't work well. So we are starting to perform this kind of analysis. So it's a work in progress.
Thank you for that. Is it possible that modified protein may not have a deleterious function, but could actually help? Is it possible?
Is it possible that there is the presence of a protein that got a missense mutation, but maybe we have to characterize this mutation, because not always we have a clearly in vivo prediction of the missense mutation, because we, with the next generation sequencing, the sequencing that we perform right now, we got variants with unknown significance. So we are studying also this kind of variation.
I think that's very important work, especially because there are so many different mutations, and each one could produce a different protein with a different effect. So it's very important. Thank you.
Thank you very much for your presentation earlier. I have just a wonder about the presentation that you talked about from Dr. Abedan, that looked into different medications, possible medications, and I don't know if I understood well that one of the deloxetine, the drug that is also used for depression, if I understood?
Antidepressive, yeah.
Antidepressive, because we also heard a very interesting presentation from Dr. Lagali that talked about the brain and endocrine function. Are you considering maybe cross-border research on if there could be like a drug that could actually work for two different things, like systemic drug, that could actually have an impact both on the eye and then the brain, or the function of the patient? Could that be something for the future?
That's a very interesting observation, yeah. Yes, systemic drugs, whether they, it's a good possibility, something to look into, certainly. I don't know if Jim, if you have any comment on that. We like to, we of course with the eye, we like to give things very locally, and if we give it systemically, we have to show that it actually reaches the eye in the amount needed to actually have an effect. Of course, then it's also going to have systemic effects as well, and those would have to be somehow measured as well, those effects. But it's very interesting too. I think the first thing would be to look at some kind of a model and see if, because of course we give mice oral treatments and systemically, so it would be interesting to look at that to see if, but there could be also, it could be a timing effect too, because there can be, only a certain window where you could actually affect brain function. Really, that's not my field, but really, that's interesting. Interesting proposal. Yeah. Okay. Thank you. Thank you very much.